go in here and attach cytoplasm. So cytoplasm is organelle, cellular organelles, which is present in the both prokaryotic, eukaryotic cells, animal, bacteria, all cells have, doesn't matter what type of the cells, they have cytoplasm. And what is this? It's just like if we look on this, this is the cell, yes? This is a nucleus, this is eukaryotic cells. This is different organelles in here show, but this is background in here, all around between these organelles. So this is it. This will be the cytoplasm. It's a jelly-like or clear fluid inside of each cell. And it's just surround all internal cellular organelle or molecules in the prokaryote. And what is the function? So this is the place of the all cellular activity inside of this jelly-like cytoplasm. Place where is signaling molecules traveling from one place to the other. Metabolic chemical reactions also can occur in the cytoplasm. So this is the main function of the cytoplasm. Cell membrane. We already talked about this, but again, let's refresh. So is a fluid mosaic membrane. It's selectively permeable. It's a double layer of the phospholipids, main components of it. And we have embedded proteins in here inside of this membrane. So phospholipids, or lipids, fats, they contain hydrophilic heads and hydrophobic tails, and they organize in bilayer, yes? And main function of these phospholipids inside of the cell membrane is a barrier. They create barriers, so it's not easy for everything to penetrate easy inside of the cell. And again, hydro mean water-philic, water-like molecules. They could be mixed with water, and hydrophobic. They do not like water, hate water, so they cannot usually easily mix with water. For example, vegetable oil or butter in the water. So on this diagram show pacemaker cells which control heart rate. And in here shows that some this is proteins in here embedded in the bilayer of the phospholipids in here. They could be bind to the molecules, such as in here show, for example, acetylcholine. It's binding, and this special protein, it's a receptor. It means it has this, as we can say in here, special site on this molecule, in here in the yellow special site. And it's this site, receptor site, it have a 3D special shape. And only one type of the molecule can be fitting in here. And after this molecule go in here and attach to this receptor site, like a key and lock mechanism it has. So only one shape can fit in this receptor. So receptor is like this. Only one type of the molecule can fit in here. And after this happening, for example, this receptor protein now can initiate different chemical reactions, and in the end, for example, this receptor cause opening of the transmembrane protein channel, and for example, potassium in this case can leave the cell, but it's only happen after, for example, some molecules attached. So this is the other example of the important transmembrane protein in here, which can initiate opening of the channels. They call receptors. So the other, what we're already talking about, cholesterol molecules, for example, which prevent stiffness of the membrane because they separate this is hydrophobic tails of the phospholipids. Glycolipids could be this on the surface of the cell molecules, which could be as a signal molecules, and we can have glycoproteins, for example, the other in here molecules attached to the protein in here part, and they could be also attached to to the antibodies or something. So they have on the membrane different glycolipids, glycoproteins, which can have additional function, depend on the cell. So let's see about other proteins which embedded in the cellular membrane and what functions they have. So I already talked about channel proteins. So they open, they create this channel in the membrane, and special molecules after channel is open can move inside of the cell. For example, it could be carrier protein. They will change, remember, shape. They can again not grabs, but they can help molecules specialized to move from one side of the membrane to the other side of the membrane. Then we will have receptor proteins, and this is what I tried to explain in previous slide, when molecules can trigger or initiate something, set of the responses, for example, opening channels, what I was demonstrating on previous slide, or release of the hormone, it could be initiated. So if one special shaped molecule attached to the other protein receptor on the membrane, after this happening, attachment to the receptor, this protein receptor will initiate several different chemical reactions, and in the end it can or open channel, or the cells can release hormone. For example, if it's pancreatic cell, for example, and it's releasing hormone, insulin. Okay, the other example of the specialized function of these transmembrane proteins could be cell recognition proteins. So on different cells of our body, we have inside of the membrane embedding protein, and they will have specialized some kind of 3D shape, and this will be ID tags. So this specialized shape on the surface of the cell, ID tag. And when our immune cells, they have ability to move around the cells, and when they come, they attached to this ID tag, they recognize it. And because of this, they say, oh, I identify this ID tag. They show me that this cell in here, our body cells, so I will not to do anything with this. But if it's moving to the other cell and this immune cells, and it will not find on the surface this specialized ID tag protein embedded in here. If it's not present in these cells, it means that now this immune cell will surround and destroy it, because it's recognized it's not our body cell. Maybe it's microorganism penetrating here. So this cell recognition protein on the surface of the membrane, very important. And if this mechanism is damaged and our own cells do not produce this as cells recognition proteins on the surface, it means it can cause autoimmune disorders, when our immune cells will recognize our cells like, oh, if our cells without this ID tags, our immune cell will destroy it, like it's foreign molecules. Yes, so this is what is the autoimmune disorders. Autoimmune, like our immune system attacking our own cells in this case. The other type of the proteins which could be located on the membranes could be enzymatic proteins. And all enzymes or enzymatic proteins, they function is to carry out different chemical reactions. They help to carry chemical reactions fast. And again, when we was talking about chloroplast, yes, and thylakoids, which made up of this stack of the membranes like coins. Again, I was talking that we have in here in these membranes of the thylakoids, we have enzymes sitting. And this is where these chemical reactions occur. and these cells can produce energy, for example, ATP molecule. Okay, next, transport across membrane. So this is our previous lecture, what we're talking about. So the membrane is selectively permeable, so not everything penetrates freely in and out of the cell. Only selective molecules can penetrate. So usually small particle or particles without charge, like oxygen, carbon dioxide, they can pass through this bilayer of the phospholipid. So cellular membrane. Water has charge, so it cannot freely move through the bilayer of the phospholipids. And because of this, inside of the membrane, we have special transmembrane protein channels, which help water to move in and out of the cell. They call aquaporins. Now, in here, we will again go over very quickly through the passive transport. So we have diffusion. Molecules can diffuse from high concentration to low concentration. So this is down the concentration gradient. We have facilitated diffusion. So this is when movements of the molecular ions across membrane help or assist by the specialized transmembrane protein channels or carriers. And on this slide below, we will see about osmosis again. So it's a diffusion of the water across membrane. And we can see in here 3 different solutions, and this is black background in here. It's red blood cells in different solution. So in here, in the number 1 solution, it's a hypertonic. Hyper means a lot. So this solution black have high concentration of the salt. Imagine we have a lot of salt in here. So much that it is much more concentration compared that it's inside of the cell. And water will follow the salt. So it means water moving away from the cell and cells become shrinking in here, smaller and smaller, damaging. This is what happening, water leaving the cell. It follows a very small salt. And this is hypertonic solution. So this is why water will move to the salt, away from the cell, and cells will shrink. Next, second in here, black background, this will be isotonic solution. So it means, iso means the same. So same concentration of the salt inside and outside of the cell. So will be the same. This is why what happening? Size of these red blood cells do not change because same amount of the water go in and same amount of the water go out in the isotonic solution. So this is particularly important to know and understand when somebody introducing IV, for example, to the patient. So any solution which is introduced in the body need to be isotonic to the blood. Same concentrations need to be osmolarity. So inside, when we introduce this solution, we will not break our red blood cells. So this is need to be isotonic. So our cells will not be damaged. And then on this, in the third condition, what we have, this is black background, is a hypotonic solution. So it means hypo means low. So it will be outside of the cell will be less concentration of the salt compared inside of the cell. So in this case, water will move, follow the salt and move inside of the cell. They will increase size and then they will be damaged, burst completely. So this is what will happening in the hypotonic solution. Water moving in and here. So again, osmosis, it's mean water moving across membrane. And in here, 3 different type of the solution and what happening with the red blood cells. Next, active transport. So it means movements against concentration gradient. So all passive transport moving along concentration gradient from high concentration to low concentration. In active transport, opposite, we're moving against. So, for example, we have a lot of sodium in here, outside of the cell. But this is sodium-potassium pump will spend ATP molecule in here represent, but it will move sodium from cell outside of the cell against concentration gradient. Outside we have high concentration of the sodium, inside we have low. But this transport is an active transport. It requires energy. We're spending energy, but we're moving against this concentration gradient. We're moving from low concentration of the sodium to high concentration of the sodium. And then here shows this transmembrane protein with this specialized shape in here, designed only for the sodium molecules to move. When it's this transmembrane protein will change shape, conformation, what will happening? Sodium now in here will leave outside. But in this case now, potassium ions from outside can move to their side in here, 3D shape, and attached in here, what's showing in this diagram now, potassium attached. And then when this transmembrane protein change conformation, potassium dissociate from this place and move inside of the cell. And it's moving against these potassium ions against concentration gradient. We have already a lot of potassium in here inside of the cell. We have high concentration of the potassium, but this transmembrane protein move it along concentration gradient, whereas we have potassium and moving even more. And outside of the cell, we have less concentration of the potassium. So, and this is example of the sodium-potassium pump, how this work. It's pumped 3 sodium and 2 potassium, and it's moving them against the concentration gradient. The other example of the active transport, endocytosis, when we're taking substances inside of the cell, endo mean inside. Example, pinocytosis, when we're moving water inside, or phagocytosis, phago mean eating. Cytosis means cell. So when we're moving some solid particles, food particles inside of the cell. Or it can, the other example, exocytosis. Exo mean we're pushing some substances, we're moving them outside. Exo mean outside of the cell. For example, removal of the waste product away from the cell. All these processes are example of the active transport. They're moving against the concentration gradient, and they all require ATP molecules. So overall, function of the cellular membrane. Number 1, barrier, isolate the cytoplasm from external environment. Second, regulate exchange of the substances, because inside of the cell membrane we have these different transmembrane proteins. Number 3, communication with other cells, because on the surface of the cells we have some glycoprotein, lipids or other proteins which could be part of the communication, one cells with the other, like in the case of the ID tags on the cells of our own body, and communication with immune cells, telling them this cell is cell of our body. Okay, so the other function of the membrane overall is compartmentalization, creating of the little these compartments. Yes, different organelles, like in here, mitochondria, nucleus, Golgi, endoplasmic reticulum. So all these organelles bounce around this membrane. And why it's important compartmentalization? Because inside of the different type of the organelles, we have different biomolecules, we have different enzymes, and we have different chemical reactions occur. In order to separate these different chemical reactions, we have membrane surround these organelles, compartmentalization. And this is example of this cellular organelles such as mitochondria, lysosomes, plasmic reticulum, nucleus, Golgi apparatus.